interact. Oh, hi everyone. This is Chad Pecot. I'm the director of the UNC RNA Discovery Center. It's my privilege to be able to introduce a couple slides on the RNA Collaborative, as well as Yinglong Miao, who's going to be giving our talk. The RNA Collaborative, as you know, is an international effort, and today is a great example of that. We got people from UNC presenting and all the way from Singapore. So I think we're about 12 hours apart. So it's a neat example that we could be, integrating and collaborating in this way. So the RNA Collaborative is humongous and growing. I think we're over 30 RNA centers around the world, many of which are represented here. We have a new logo, so we'll be replacing this one, but it's really neat to be able to just see how many people around the world are joining this, and many of you are on today. I do want to remind you, this is actually a recorded session, so you'll be able to get the recordings. We request no screenshots or photos or recordings of your own. We will provide the recording to you through a YouTube channel. All right, so I'm going to be the first to introduce our first speaker. So Dr. Yinglong Miao, he is an associate professor in the Department of Pharmacology and Computational Medicine at UNC, Chapel Hill. He develops novel accelerated simulation methods and applies these methods in advanced simulations of biomolecules and drug discovery. His lab currently focuses on simulations and drug discovery of important membrane proteins and RNA. You'll be able to see a lot of that today. He's received several distinguished international awards, and he's currently the editor-in-chief of NPJ Drug Discovery Journal. So be nice to him if you submit papers there. All right. I'm going to let Yinglong take it from here.
Thank you very much, Chad, for the nice introduction. So can you see my slides?
Yes.
Great. Well, good morning, good afternoon, good evening, everyone. I realized we have people from different time zones. So thank you very much for being here. So my lab develops new methods to study important drug targets. This include like membrane proteins, like G protein coupled receptors or GPCRs. RNA is relatively new direction in our lab, but I think it has enormous opportunities for submissions and drug discovery. So I'm very excited to talk about it. And today, I will tell you a couple of stories about how small molecules bind RNA. Well, before I dive into the research projects, I want to thank the people who make this possible. So my lab moved from University of Kansas or KU to UNC in 2023. So that's about 2 years ago. On the top, you look at the two more recent photos at UNC. At the bottom, it was our last photo before we left the KU. Now today, I will talk about the projects managed by Sana, who has been an expert in our lab. The other thing of funding the computational resources. I also want to thank our wonderful experimental collaborators from Jinxin Wang's lab at the University of Chicago. Jinxin, his postdoc Zhi Chao, and a graduate student Shileka. I also appreciate the discussions with Professor Eric Holmstrom from KU. For this audience, I think we will know the RNA structures have emerged as new, exciting therapeutic targets. So these include MIs for the proteins that are difficult to drug. They also include the non-coding Is that are involved in different diseases. And we have different methods to design small molecules that can bind the I. You can check out these nice reviews from experts in the field. On the other hand, molecular bindings to I is known to be slow. If you look at the ligand binding rates, like K-on, they fall like far below the diffusion limit. They are even much slower than the protein ligand binding rates. Then, how do the small molecules bind RNA? I think this is a very challenging question for both experiments and the submissions. For experiments, it's rather very hard for us to reach the time and the space scales in order to look at the ligand binding directly. For submissions, we use a tool called a computational microscope. For this, we talk about the molecular dynamics or MD submission. MD is based on chemistry, physics, biology, and computer science. In MD, we solve Newton's equations for all the particles in the system. Then from MD submissions, we can see very interesting biological processes. On the left, we look at one example MD submission. It captured a protein folding process. So starting from a long stretched polypeptide, the system here can fold the protein into the native X-ray crystal structure. So this protein is fairly small. It has only like 3 alpha helices. As you watch the protein conformation, we can figure out the pathway or the mechanism of the protein folding process. On the right, we look at another MD submission. It captured the protein drug binding process. As we track the drug molecule, it can search the protein surface and bind the target site. Well, as you can see, MD is very powerful computational microscope. And with this microscope, we can essentially watch the biomolecules in motion. However, MD has a limitation. Even with our fastest supercomputers, we normally run MD submissions up to microseconds or tens of microseconds. On the other hand, many biological processes, like unlinked binding and unbinding, could take milliseconds to seconds, even longer timescales. So we have a large gap between the timescales of our submissions and those of biological processes. Then we want to develop new methods to speed up the submissions and bridge the gap. Well, now, if you look at the problem, our proteins now have a free energy landscape. So they have different low energy states separated by high energy barriers. Well, now, like in our very short MD submissions, it's very hard for the proteins now to jump out of the low energy wells and sample different states. Well, with that in mind, as Chen mentioned, we developed new methods to address this challenge. Today, I would focus on 2 important questions. First, how do we speed up the submissions? Second, how do the small molecules bind I in our accelerated submissions? For the first question, we have been developing methodology over the past 10 years. This is called GAMD, or Gaussian Accelerated Molecular Dynamics. It's one of the enhanced sampling techniques. In GAMD, we add harmonic boost potential to reduce the system energy barriers. As I showed you, our proteins now have different low energy states, like bound, unbound, folded, unfolded, while in between, they have high energy barriers. Now, given reference energy E, when the system potential drops below that, we add harmonic boost potential. So this is our formula. As we change the harmonic force constant K, we can raise the low energy wells, reduce the energy barriers, then we can speed up the submissions. Well, in GAMD, our boost potential norm has no range. It shows a Gaussian distribution. Then we can apply cumulative expansion to the second order, or just a Gaussian approximation to reweight the submissions. So we want to calculate the original free energy. It's the modified minus the boost potential cumulants. For the cumulants, the first order the average, second order the variance. With this, we can calculate the accurate free energy profiles from GAMD submissions. Well, in comparison, conventional MD is like hiking. We walk step by step to climb mountains. I think many of us, including myself, really enjoy hiking. It's fun, but can be very slow. So every step of walk in MD submissions would be 1 over a few femtoseconds. That's 10 to the minus 15th of a second. Even as we walk billions of steps, we reach only microseconds. While in GAMD, we fly on helicopter, we lift ourselves, we have much higher potential. Then we can fly over many mountains very quickly. We can enjoy many more views. Then by running short microsecond submissions, we can capture long timescale events, like over milliseconds. We have shown this in a number of previous studies, especially for GP cells and CRISPR-Cas9. Then we call GAMD a time-accelerated computational microscope. It has been coded in popular MD packages in our field, including MBER, NumD, OpenMM, Genesis, Tinker, and also Discover Studio that's used in pharmaceutical industry. People have used the GAMD to study protein folding, conformational change, ligand binding to different proteins in the eye, interactions between proteins and all the other biomarkers, the peptides, proteins, carbohydrates, and nucleic acids. It also works for allostery and exploring different strategies for drug design. Based on GAMD, my lab has also developed new selective GAMD algorithms. These methods include ligand GAMD, peptide GAMD, and the protein-protein interaction GAMD. By adding selective boost potentials, we can speed up our submissions by thousands to trillions of times. Then by running only microsecond submissions, we can capture both the dissociation and binding of like small molecules, peptides, and proteins. Then we can calculate the binding thermodynamics and kinetics. So these include the binding for energy delta G and also kinetic rates, K on and K off. While these are critical parameters for therapeutic design of the drugs, peptides, and antibodies. Because I use LIGAMD to study ligand binding today, so I will tell you more details here. In LIGAMD, the basic idea is to add a selective boost on the ligand. This is for the non-bounded interaction energy between the ligand and the receptor, like the protein in the eye. With this boost, we can push out the ligand and speed up the ligand dissociation. In addition, we add another boost to the rest of the system. That includes ligands in the solvent, so you can speed up the ligand diffusion and help ligand rebinding. So these are our new similar methods. Next, we'd like to apply them and say how small molecules bind RNA. Our first target is the survival motor neuron 2 or SMN2 pre-MI. As we all know, if there's any mistake in the splicing of exon 7 in SMN2, that could lead to spinal muscular atrophy, SMA. This is a very bad genetic disease in the babies. Back in 2020, risdiplam was the fastest splicing drug approved by FDA for treating SMA. So this is the chemical structure. And according to the previous studies, people suggested there could be two potential drug binding sites in SMN2. One is formed at site 1. It's between U1-SI and the SMN2. So it's formed by double-stranded line. The other is at site 2. It's formed by like a single-stranded line in only the SMN2. In one MR structure published in 2019, people showed the drug would bind at the site 1 formed by the double-stranded line. But we don't know like how the drug could bind a single-stranded line in at the site 2. Yeah, with that, we worked with Professor Jin Xiwang's lab in medicinal chemistry. So we set up GAMD submissions to look at the ligand binding. We have this on a target. Initially, we put the ligand far away in the solvent. We have explicit water molecules and ions. As you run the submission, the ligand could diffuse and bind very quickly to the ion. It can induce the folding of the ion structure, and they can rearrange the conformation. At the end, they form a compact, very beautiful complex like this. And we observe the ligand binding to both RNA and the DNA. For the first time, we discovered a novel ligand binding pocket formed by two sequential GAAG loop-like structure. So the ligand could form pi-pi stack interactions with the surrounding nucleotides. This binding conformation was quite consistent with the MR spectra. But I also noticed one difference. So if I look at this nitrogen N4, it's exposed to the solvent in case of DNA. but I can form a hydrogen bond with a 2G in the RNA structure. Then we checked this in the SAR structure activity relationship studies. So in the experiments, our collaborators changed this chemical group on N4. When we have a bulky, like a much larger chemical group, it can reduce the ligand binding affinity in the eye, but it's no effects on the binding to DNA. So I think yeah, that's quite consistent with our game prediction. Also found that this D and E bisected ring is critical for ligand binding. If we change it to a smaller like a phenyl ring, it can reduce the binding affinities, and it can even abolish the binding. But if we put it back, it can restore the ligand binding affinities in case of a 10 and 11. So our KMD predictions agreed pretty well with the SAR and MR experiments. And combined, yeah, we can figure out this dynamic mechanism for the binding of the drug to single-stranded line in SMN2. Well, more recently with the COVID, also look at the ligand binding to I in SARS-CoV-2. So for SARS-CoV-2, people find the biological G in the stem loop 5, SL5. So SL5 is quite conserved in the virus for replication and infection. And with this target I, our collaborators in Jin Xuang's lab synthesized about 70 coumarin derivatives. So given these compounds, we'll look at the binding to the target I. And in the SL5, we also mutated this nucleotide G to A, U, and C. And we find this binding trend. The most preferred binding would be for G, followed by A, U, and C. The data here are very exciting, but we don't know how the compounds would selectively bind the G bulge. Then similarly, we set up set up KMD submissions. Look at the ligand binding. We have this target I. Initially, yeah, we have the ligand in the solvent. It can diffuse and it can actually bind fairly quickly to the eye. We found it can bind the minor groove. That was surprising in the beginning. We also noticed it can interact with the G bulge, like G24. If we track the distance from the ligand to the G24, initially we had high fluctuations, but gradually it can level off and stay around 5 angstroms. So have a good binding, even just in the very short 70 nanosecond KMD equilibration simulation. Now we move down to run much longer KMD production simulations. These are for 1500 nanoseconds. We have 3 runs. And in these simulations, the ligand form the close interactions with the G24 and the C12. So the binding is pretty stable. We take these 2 distances as our reaction coordinates. Then we can calculate a free energy profile. In this free energy profile, we can figure out a number of low energy states, like a bound and the intermediate. In this bound state, the ligand could form ionic interaction with the phosphates from the backbone, from a U25 and the G24. It can also form a very important hydrogen bond with the G24. And I think this is critical for the selective binding. And the ligand also form the peptide stack interaction with the C12. Then we added the KMD submissions on a couple of inactive compounds. We thought this could be requested by the reviewers. It's it can serve as a negative control. So here I would show you one of the examples. We have this inactive compound SM64. The experimental KD would be 50 micromolar, so it doesn't really bind. Then yeah, with the KMD submissions, the ligand or the compound can get to G24 and the C12 very briefly, but for most of the time, I mean, it's far away by more than 50 angstroms. Similarly, if we also calculate a 2D French profile, we find only the unbound state. So this is the confirmation. The ligand is, the compound is pretty far away from the target site. Well, now if we go back to the potent ligand C30, we find organic prediction was very consistent with MR spectra. So in the experiments, our collaborators added a number of MR labels in different nucleotides. So these are highlighted in spheres here. And then we titrate the ligand at the different concentrations. We look at the change in the R2 relaxation rate. We find the largest increase in the number of nucleotides. These include G9, A10, U22, C23, and the G24. And if you look at all these nucleotides, they're highlighted in purple. So they're found in the target site as we predicted from GMD submission. So again, like GMD submission and the prediction agreed very well with the MR experiments. Combined, we found like this ligand could bind the minor groove in the SL5. And I think we figured out this like a mechanism, dynamic mechanism for ligand binding to the SARS-CoV-2. And our paper just came out in last month. So if I'm interested, you can feel free to check out the paper for more details. Well, last but not least, we also apply ligand submissions to study the thermodynamics and the kinetics of ligand binding. We look at the binding of a small molecule, thorphalin, to the aptamer. So in this MR structure, the ligand binds at the target site, as shown here. Then we also added another 2 ligands in the solvent. Then we run like a multiple 2.5 microsecond submissions. From our submissions, we calculated the RMSD, root mean square division of the ligand compared with MR structure. As you can see, the ligand could bind and dissociate multiple times in the submission. With enough sampling, we can also calculate the ligand binding FNH and the kinetics. At the same time, our collaborators can measure the ligand binding, like a free energy and the kinetics from SBL experiments. In comparison, our estimates are pretty accurate. Now we can go into the submissions and figure out more mechanisms. So in the submissions, this C27 can move out and in during the ligand binding process. You can see the different conformations. Then we can calculate its SASA, solvent accessible surface area. We'll take this as one of the reaction coordinates. We can also take the ligand RMSD as another reaction coordinate. And then from the free energy profile, we can figure out a number of low energy states. These would include the bound for the ligand, intermediates, and also unbound. We have the in and out for the C27, the nucleotide conformations. And you can check out these pictures for the different conformational states. Well, as we track the ligand, we find the multiple pathways for ligand binding and dissociation. For the binding, we have like 3 different pathways. If we take this as the front view, in the first pathway, the ligand can bind from the upper right direction. So you can get close to like a G26, and then finally the target site close to C27, G24, and A28. In the second pathway, it goes below G26 from like the right side. In the third pathway, it goes from like the lower left region. So you can get close to A5, C30, and then finally the target site. For the dissociation, the main pathways come from the reverse of the first binding pathway. With this, we can also calculate the ligand binding K on and K off. For sake of time, I will not like talk about all the calculation details, but I would say like in comparison, they agreed pretty well with the experimental data. Well, in summary, I told you a couple of stories about both methods and applications on ligand binding. So I have developed a methodology called GMD. We have created a new time-accelerated computational microscope with our recent selective GMD algorithms like GMD, we can speed up the submissions by thousands to trillions of times. With only microsecond submissions, we can calculate both the binding thermodynamics and the kinetics of a ligand binding. For SMN2 and SARS-CoV-2, our gamma submissions can help us to figure out the pathways and the mechanism of ligand binding to a single strand line. Our gamma predictions were quite consistent with AMR and SAR experiments. And then with the ligand submissions, we can also capture both the binding and the dissociation of small molecules to an aptamer. We can estimate the binding thermodynamics and the kinetics. Finally, I'm as Chen mentioned, I'm editor-in-chief for the new Nature portfolio journal, MPG Drug Discovery. So publish research articles, reviews, comments, perspectives, and protocols. Right now, we have a number of active collections, and we have one upcoming for drug discovery on RNA and RNA binding proteins. So if you have manuscripts, that could be a good fit. We're looking forward to submissions. So thank you very much for time. Yeah, I'll be happy to take your questions.
Great job, Yinglong. I'm not seeing any questions coming in just yet. Actually, here you go. Here's one right now. Great talk. I was wondering if the approach can capture large conformational changes during ligand binding.
Indeed, that's a great question. We can go back to some movies. Let's see. Yeah, we can look at this movie, right? So initially we have this kind of like a extended RNA sequence. When you have the ligand binding, that can induce like folding of the RNA structure. So you have like a very large conformational changes. This is for SMN2. And we can see the same thing for, let's say, like the RNA aptamer, the model system. Not here. Yeah, this shows the different conformations. So you can see, yeah, especially this C27 as I highlighted, yeah, can move in and out, and you have like a very large conformational space you want to sample during the ligand binding. Hopefully, yeah, that can answer the question.
That's great. Yeah, I think just for sake of time, I'm going to give it over to the next moderator and next speaker. Thank you very much, Yinglong. It was a great talk.
Thank you, Chad.
Okay, let's move on to the second speaker. Is there any slides to show, or I directly introduce?
I will share the slide.
Okay, thank you. Hello, everyone. My name is Polly Chen from Cancer Science Institute of Singapore, National University of Singapore. It's my great pleasure to introduce Dr. Jessica Ho, assistant professor at Duke-NUS Medical School. Jessica is a highly active and respected member of our local RNA community. Especially the NUS RNA supergroup. You can see the new logo at the upper right corner. And her lab is dedicated to understanding the epigenetic and transcriptional mechanism that shape cellular response to influenza A infection. Specifically, her research explore host RNA splicing changes, chromatin control of the host response, and the related area. Without further ado, please join me in welcoming Jessica as she delivers her talk. Thank you so much. Thank you, Jessica.
Thank you. Let me start share. Is it working?
Yeah, but it's not on the display mode.
It's not on the display now. Yes?
Yeah, now it's okay. Yeah,
Okay,
thank you.
perfect. Okay, thank you. Okay, hello everyone. Thank you everyone for taking the time out to come to this wonderful RNA collaborative seminar series. Before I start, I wanted to thank the organizers for giving me this really wonderful opportunity to share some of my work. So today, I'd like to share with everyone a bit of my work on alternative splicing. So to put things in context, I'm particularly interested in understanding how cells regulate their transcriptome and their epigenome, especially in response to environmental stress and transitions between cellular states. So the reason why I'm interested in this aspect, like the transitions from healthy to disease states, is because during these transitions, both the transcriptional and epigenetic machinery must be rewired. And this rewiring is actually required to make sure that the cell can express the appropriate genes. It needs to be it's important for making sure the cell has the correct gene expression amplitudes, the correct durations of gene expressions, as well as the correct kinetics of gene expression. So what genes go first, what genes go next, what genes goes last. And because of this very important control in gene expression, we are very interested in understanding how the different mechanisms maintain this expression dynamics. And I'm particularly interested in understanding how chromatin organization and processes and RNA processing, more particularly in splicing, impact the cellular response to both infectious disease and a little bit in cancer. So as I'm sure most of this audience already knows, splicing is a very important means for a cell to expand the coding capacity of its genes. So in eukaryotes, our genes are organized into very distinct structures, being exons and introns. And during the process of transcription, introns can be excised in different combinations, leading to different combinations of exons being put together. So this leads to the generation of multiple splicing isoforms that arise from a single gene. And this is a very important way for the cell to diversify its coding repertoire. Because if you have an extra exon in a particular gene, you can gain functions in the gene. So for instance, in this protein that is translated from this transcript, you have an additional pink domain that could change the protein function because this piece of amino acid could interact with other genes, other proteins, or it could encode a new domain that could be important for the cell to make its function. So as a result, splicing is actually very highly regulated and very tissue specific. So one critical aspect of this system is that the investigation of splicing can be very useful for studying cellular state transitions. And these are also particularly in regions that are not observable by gene expression alone. And what this means is you can see new cellular states or subpopulations that you don't necessarily see from just looking at DEGs. And one very nice example of this comes from a paper from the Yo lab. So here I show protein kinase M. So this is a differentiation of neurons from iPSCs all the way to motor neurons. And if you look at the gene expression profile of protein kinase M from iPSCs all the way to the mature neurons, you see that expression of PKM actually doesn't look that much different between the three cellular states. If you stop here, you would say that PKM is not very important for neuronal differentiation. However, if you start taking splicing into consideration, the picture looks a little bit different. So for instance, here they looked at exon 9 inclusion of PKM, and you can immediately appreciate the difference between these two plots. You can see that iPSCs don't really have PKM isoforms that have exon 9, whereas in motor neurons, you have PKM isoforms that express exon 9, showing the big importance of looking at splicing when we look at cellular functions or transitions between cellular states. So I think this is one region that it would be very important to look at when we look at stress responses or we look at disease to healthy to disease transitions. So which brings me back to the central hypothesis in the lab. So what we are trying to do now is, so if splicing is cell state and tissue dependent, then perhaps one prediction from this observation is that maybe cells under stress, either during cancer, oncogenesis, or during infection, could actually be very specifically reliant on very particular splicing regulators or events, and for them to maintain or prolong that cellular state, or to exit from that cellular state. And if this is true, then maybe such events could present very good therapeutic targets for us to attack if we know more about them. So as a very short example of this concept, I wanted to talk a little bit about some of my previous work. So here, let's consider the role of splicing factor HNRNPM in cancer. So previously, I found that HNRNPM was a dependency factor in prostate cancer. So if you look at the TCGA database, you can see that HNRNPM is highly expressed in tumor tissue and not so much in the adjacent normal tissue. Patients with high levels of HNRNPM expression have a much poorer survival prognosis compared to patients with less HNRNPM expression. So HNRNPM looks like it's a very important gene for the regulation of prostate cancer growth and survival. So we decided to look at this gene, and to cut a very long story short, even though HNRNPM is a very ubiquitously expressed gene, so it's expressed in all tissues in our body, the knockdown of HNRNPM surprisingly did not result in global changes in splicing. So when we did the alternative splicing analysis for HNRNPM knockdown cells, it turns out there were maybe about 200 genes were differentially spliced when you knock down HNRNPM. And of these 200 genes, only more than half of them were genes that displayed changes in skip exon events. So in this, when we looked at it further, we saw that most of the genes that had changes in alternative splicing ended up adding new exons to their transcript. So, and you can see this with the splicing gels here. So the first lane is the scrambled treated cell. So you can see the usual transcript that the prostate cancer cells express. Upon knocking down HNRNPM with 2 independent shRNAs, you can start seeing the expression of a new transcript isoform that is much larger than the original isoform. And some of these switches were very big. You could see almost 90% switch in splicing in some of the events that we picked up. You can see here. And then some you see a little bit less. When we look at DEGs, I don't show the data here, but we also don't pick up many DEGs. So it looks like HNRNPM was a very specifically impacting splicing levels in the cells. When we looked at the gene ontology of these genes, many of these genes were very important for cellular function and for regulating transcription or chromatin environment in the prostate cancer cells. So these include many chromatin regulators like histone-modifying proteins and histone-modifying proteins. So looking deeper, the reason why HNRNPM has such a specific effect on splicing was because it was bound to and regulated splicing under very specific conditions. So we did E-clip analysis, and we found that HNRNPM was actually very specific for GU-rich repeats in the introns of its target genes. When you then look at the location of these GU-rich repeats, you find that HNRNPM was very highly enriched at the flanking introns of the exons that it controlled. So if this is an exon that's regulated by HNRNPM, the -1 and +1 intron always had the most number of HNRNPM binding sites in the transcript. And these introns, the -1 and +1 introns, always happen to be much longer than any other expressed intron in the cell. So because of this architecture, HNRNPM was able to exert its very specific effects on the pool of 135 transcripts that it regulated. And when we look deeper, of these target genes, we've decided to focus particularly on EED protein. So EED is a member of the polycomb repressive complex 2 that deposits lysine 27 trimethylation on histone H3. So what we found was that if we knock down HNRNPM, this resulted in a 75 nucleotide exon inclusion in the EED transcript. What this did was it added this 25 amino acid peptide that in-frame into the EED protein. Now, 25 amino acids doesn't sound very long, but it turned out that these 25 amino acids had quite a big impact on the EED protein. So EED protein is actually is a beta propeller structure, and it comprises of 7 WD repeats in the protein. And what this 25 amino acid peptide did was it disrupted the beta sheet in WD repeat 5 of the EED protein. And this disruption was predicted to destabilize the entire EED protein. And indeed, when we did the Western blots looking at HNRPM knockdown using a splice switching oligo, what we found was that H3K27 methylation was actually much reduced in the HNRPM knockdown cells compared to scrambled treated cells. Similarly, if we use a SSO to force this splicing event, the EED SSOs, we also could reduce H3K27 methylation levels equally. More importantly, we were also able to show that we could phenocopy HNRPM loss by forcing prostate cancer cells to express the misspliced isoform of EED. So by using these splice switching oligos, which force the inclusion of the EED exon that we were interested in, we could show that we could reduce cell proliferation in prostate cancer cell lines. So these are the 2 EED splice switching oligo treated cells. Compared to the scrambled treated cells, they have 50% and 40% reduction in growth. Similarly, you see that HNRPM SSO, even though the switch was able to achieve the same level of also achieve knockdown of the growth. So I think the key of this was that I think here we were able to show that in addition to knocking down splicing factors, we can actually also manipulate key pathogenic splicing events for therapeutic interventions. So to quickly summarize this part, I've showed that HNRPM regulates very specific subsets of genes and is an important regulator of prostate cancer cell growth. I've also showed that alternative splicing events suppressed by HNRPM, such as EED, are quite detrimental to cell growth and could potentially be used as therapeutic strategies by using splice switching oligos. And I think this basically comes back to our initial hypothesis is that cells under stress can really be specifically reliant on particular splicing regulators or events, and then such events can present good therapeutic targets in the downstream. So how does this link back to infection? So in the context of cancer, there's a very chronic long-term rewiring of the cell's epigenetic and transcriptional status, and that could potentially explain to why splicing factors like HNRPM become weak points for cancer cells in the long term. However, what about situations like an infection where transcription and epigenetic rewiring occurs in much shorter timeframes? Then I think the question then becomes is how actually important is alternative splicing in this context? And I think this is the question that we are trying to answer in the lab right now. So to address this question, what I wanted to do was to really investigate how acute viral infection impacts alternative splicing during infection. And the virus we picked to work on was the influenza virus. And I'm sure everyone knows this is a very globally significant pathogen. WHO estimates there are nearly 3 to 5 million severe cases every year and nearly 290K, 650K to 650K deaths that can be attributed to influenza every year. A main problem with this virus is that it mutates very quickly, which means vaccines have to be updated annually. There's also increasing gain of drug resistance within the viral populations. So overall, I think the idea here is we do need to find new avenues to combat this virus, and learning more about how it affects the whole cell could be one important strategy for designing new therapeutics or prophylactics against the viral infection. Influenza is also a very special virus among the RNA viruses because it's one of the very few RNA viruses that replicate in the nucleus of the whole cell. It doesn't encode its own capping machinery, which means that it makes mRNA through a process called cap snatching. So here it steals the caps from the host transcripts to put a pen onto its own, and in doing so, it makes its own viral mRNA. And because of this cap snatching function, influenza viral proteins have to interact very closely with the host RNA polymerase II. And in doing so, it completely disrupts the way the host polymerase initiates, elongates, and terminates on the chromatin. And in addition, this is going to lead to alternative splicing changes. Another aspect is that the virus itself also requires host machinery for alternative splicing of its transcripts NS2 and M2. So we were interested in understanding how influenza impacts alternative splicing in the host cell. So there have been some studies on this in the past, but many of these studies relied on transformed cells like A549 cells. So these are immortalized or cancer cell lines, and short read sequencing. And since splicing is such a tissue-specific or cell state-specific event, we were wondering if maybe it would be interesting to characterize alternative splicing in primary human cells to get a more physiologically relevant picture of what is happening during infection. The other aspect of the previous studies was many of them relied on short read sequencing, which, while very powerful, cannot capture the full transcript repertoire and the combinations of exons during infection. So to address this, we set up the following experiments. We first infected primary lung epithelial cells we got from donors. And what we did was we took these cells, infected them with influenza A virus, isolated the RNA, and then we split this RNA into two pools, one for short read sequencing using Illumina, and another for long read sequencing using PacBio sequencing. So the reason why we did both short and long read sequencing was because of the pros and cons of both. So short read sequencing is powerful in the sense that it can achieve very high depth of sequencing, meaning it's very easy to quantify particular exon junctions and splicing events in these reads, but you can't assemble full transcript and gene models. Short read sequencing has lower depth of sequencing, but in doing so, you're able to sequence the entire transcript and have no confusion about which exon combinations are occurring in these transcripts. So we took these two sequencing, used both the sequencing reads to generate virus and tissue-specific transcriptome and gene models. And doing so, we took these and then used for differential gene and isoform expression analysis. The tables on the right show the raw data from the PacBio sequencing analysis. We were able to get about 3 million reads, average of 3 million reads per sample. We had triplicates. And then most of them fell within the 1 kb to 6 kb. We were also able to pick up very long transcripts that were greater than 7 kb long. But in general, we were able to capture most of the transcripts, the full-length transcripts in the infected cells. After several quality control steps, we arrived at about 42,000 different transcripts that passed all our filters. We removed anything that was incomplete. We also removed any new monoexonic genes that we found. And interestingly, what we found, and then most of these transcripts were also, we also had multiple isoforms per gene. Some were only one isoform per gene, but I think about 60% of them were multi-isoform genes. So what was interesting about our data was when we compared our new transcriptome with that of the GenCode the HG38 database, we found that we could only attribute 2/3 of them to previous annotations. So this 27,000 transcripts were represented full splice matches from the current GenCode database. And another 1/3 of them represented transcripts that hadn't been annotated before. So we had about a quarter that were novel isoforms with new combinations of known splice sites. So you can see here, you miss this combination of exons. And about another 1/10 of them were novel, not in catalog transcripts. So these were new isoforms of genes that had at least one new splicing site that didn't exist before in the previous GenCode database. So these types of transcripts were evenly distributed across all different transcript lengths, meaning that we didn't see a particular length bias when we did our data. And then as an example, I show you one of the raw data that we get from this sequencing, the long-read sequencing data set. So this is the MX1 gene, which is only expressed upon viral infection. You can see in the mock infected cells, we were able to pick up two transcripts from MX1, and then upon infection, you see increased number of MX1 transcripts. And if you look at the transcript models that we were able to generate from this gene, below in blue are the genes that have been previously annotated in known databases. And then in pink are the new transcripts that we picked up in our analysis. And you can see here this nice little pink exon that you can see this little bump here that we picked up in our transcript, but wasn't annotated previously. So we're very excited about events like this. We're trying to validate these new events and see how they could impact the way genes like MX1 interact with the virus. Using our new gene models and transcripts, we perform differential splicing analysis. And as expected, we do see a global change in alternative splicing in infected cells. So many of these included new exon and intron retention events. But the bulk of them actually was surprising because they represented new use of alternative transcription termination sites as well as alternative start sites. If we dive deeper into the data, we find that genes that were changing most in transcript usage were actually not necessarily correlated with gene expression. So in this graph, the x-axis shows you gene expression. So in this half of the graph, this is highly expressed, and this is significantly downregulated with infection. The y-axis shows you the level of changes in isoform usage in the between infection and mock. So, and then the red dots represent transcripts that show significant isoform switching. So you can immediately appreciate that the majority of red dots actually do not fall within the differential gene expression quadrants of the graph, meaning that it's essentially what I alluded to earlier with the PKM example, that gene expression only shows a very small part of what's happening in the cell during a cell state transition event. And actually the bulk of the story is actually under the surface and happening in terms of alternative splicing. And indeed, if we do gene ontology analysis of the alternatively spliced transcripts, we see that a lot of them are related to stress response, stimulation, defense. And so now what we're trying to do is really validate each of these different transcripts and really try to clone the isoforms to really understand what each thing is doing in the system. So I think to really summarize, we have established both an experimental analysis pipeline to study alternative splicing, and we have chosen to do this in the physiologically relevant cell model of infection. I think we've shown that there are many alternative splicing changes, and we're currently validating and following up on this. So to end my talk, I just want to thank all my lab who did much of the work on the alternative splicing, and also all the people who have contributed to both this story as well as the previous story of HNR and PM. And I'm happy to take questions.
Thank you, Jessica, for the wonderful talk. We have one question in the Q&A box. I just read out for you. What determines the alternative splicing variability to make different isoforms of PKM in different stress conditions? And for example, PKM2 is highly expressed in case of tumor.
This I'm not too sure because this was a paper by another lab. I believe that there are different expression of different splicing factors. So maybe I think you could try checking that paper from the Genios lab.
Oh, there's another question coming. Did you ever check how does it look like for any other virus? How many of the changes are driven by the infection itself? and how many by the virus. Very interesting work. Yeah, that's the very positive.
That's a really interesting question. I'm also very interested in it. There have been a couple of other papers looking at interferon, how interferon treatment and or poly-IC treatment impacts alternative splicing. So we're still trying to compare our data sets and see what it looks like. The problem is, I think a lot of these other data sets are in different cell lines and in different tissues. So I'm also not sure how much overlap there will be. So we'll see how it goes. It's a very interesting question.
Okay, you got more question. For differential isoform expression during influenza infection, do you see any overrepresented motifs that are present?
So far, we haven't noticed anything, but it's possible that the virus has a protein NS1 that also modulates splicing. So it has been shown that it can impact several splicing factors. So I feel like it will be a global change in splicing, but we will look at it as well.
Thank you. Another question, very interesting work, and how much intron retention is there as compared to alternative usage of exons? And does it suggest some sort of a generic splicing inhibition upon viral infection? Or do you think it's a change of splicing mode?
So I think we can see this. So we didn't pick up too many intron retention events compared to alternative exon usage. I feel like this may be a more generic splicing inhibition during viral infection, but we still need to do more work to answer that question.
But I also have a follow-up question. So usually I think intron retention, is it relatively harder to be detected if you're using RMAT, so those standardized pipeline?
Yes, but with RMATs, I think RMATs is slightly more biased towards exon skipping
Yes,
events.
that's it. Yeah,
It's
I
a bit
think so.
harder to pick up intron retention events, but we are using long-read sequencing, so theoretically we should be able to pick up some
I see.
intron retention events. But so far we haven't seen too many, but it will be interesting to look further. It's also possible that maybe the time point of infection we chose is a little bit too early or too late. So I think it would be nice for us to look deeper into the kinetics of this infection.
So we still have about 2 or 3 minutes, so let's pick up another question. What is the relationship between AS and ATTS? Are the two processes related or independent?
This is a very good question. I think it's a bit complex, I think. So the alternative splicing can change polymerase kinetics can impact alternative splicing, and if you prematurely terminate the polymerase, of course you will change the way splicing is occurring. I'm not too sure how we can show that with our data set though. It could be interesting. The other factor is that the virus itself also sequesters termination factors like CPSF73. So maybe part of it we're seeing is because of the virus and not directly because of changes in the host splicing machinery.
Okay, yeah, thank you. I think still 1 minute left, so I actually have one question. So in iPS cells, do you sort of expecting there's less splicing events being detected in general, like those undifferentiated cells?
To be honest, I think you will have more splicing events in iPSCs than differentiated cells because, or maybe it depends on the differentiated cells, but in the context of like in the context of neurons, when we previously looked at transcription in iPSC compared to neurons, to differentiated neurons, we see that the neurons are actually a little bit more transcriptionally quiescent than the iPSCs. So I think the chromatin is a bit more structured in the differentiated cells compared to the iPSC. So maybe there's a lot more
see.
transcription happening. Yeah.
Okay. Thank you so much. Due to the time limit, so please allow me just on behalf of this thanks everyone. Yeah, thanks everyone for joining the talks, and thanks Jessica and Yinglong again for giving very nice wonderful talks. Yeah, very happy to be part of this community.